Hi. Yeah, good morning. Yeah, Vishalini, Admiti, Akshaya, Mithula, Sridaksha, Nivedita. Good morning. Can you hear me? Can you hear me, guys? Yeah. Okay. Good morning. Good morning. Good morning. Okay, one more minute. What is this? Hind 2, right? Hind 2 specifically cut at this DNA sequence, right? Six base pair, specific six base pair cut it will make. So like that, those specificities you need to focus, because they may ask you in match the following or section column A, column B, and they give sequences, or they may give what are the functions of this restriction endonucleases. So this is the point where you need to focus. And coming to the next one, that is idle, right? So today we will be discussing about what is the next tool, that is cloning vectors. So yesterday I gave a little introduction about cloning vectors. So it needs cloning vector means it needs to have certain certain aspects, certain criteria, certain features for that matter, right? So what about others? Still, I've seen even in chemistry class only 11 members are there. I think you guys are so confident in these chapters. Huh? Please do join. Okay. So let's move forward. We can't waste time for others. No, we have to value the time at least. Whatsoever. Right. So let's see the features of cloning vectors. Right. If you see the cloning vectors features, right, it has to contain certain aspects, right? Certain features it needs to contain. Like for example, origin of replication (ORIs). So ORIs of, so if you take this ORIs, origin of replication is one of the feature of the vector. Okay. And then we have a selectable markers. Origin of replication we talked about yesterday. Yesterday we talked about origin of replication, right? So where exactly this where the replication starts, the sequence where the replication starts, the ORS needs to be there in the cloning vectors, and then we need to have selectable markers. That selectable markers which we can really distinguish which are transformants and non-transformants, right? For example, if you see E. coli, the selectable markers for E. coli includes the genes encoding for the resistance to antibiotics. For example, ampicillin is there, chloramphenicol is there. So, if you provide these drugs, right, these antibiotics in the medium, and you try to grow this naturally, what happens is they confer resistance to these antibiotics, and they live, right? So how do we screen, right? How do we screen? How can we say that these microbes got transformed? So based upon this markers, so we are saying these antibiotic resistant genes are as an markers. It means once we introduce our target gene or desired gene or alien gene, piece of gene by rDNA technology, right? The 6 steps we will be going to deal the next 5 minutes. So once we introduce in this resistance genes area, what happens is sequences. For example, you have right. Consider that this is a vector DNA molecule. Okay, this is antibiotic. For example, if you consider this, this is okay. Yet people are joining or what? Yeah, Shamili, Gina, Priya, you guys has to be on time. Okay. Yeah. So we are discussing about the features of the cloning vectors. So first one we discussed about ORIs, and then we are talking about selectable markers. If you consider this is an plasmid, right? This plasmid has some selectable markers like this. Are you able to see? Are you guys able to see? Yeah. Yeah. So if you consider this is an vector, right? The plasmid. Okay. So this plasmid has this cloning one of the selectable marker, that is, let's assume let's assume this piece, right? This piece, this white piece. One minute. Okay. Now if you can see this, assume this is a plasmid, right? So this is a plasmid, and this is oops. So this is a selectable marker. For example, this piece of white color sequence here. In this, assume this is a antibiotic resistance gene, ampicillin or chlorphenicol or tetracycline, whatever. So in PBAT 322, you will have this selectable markers. So now what we are doing is we are inserting our target gene. Assume this is a target gene. Okay, so this is a target gene we are introducing within this sequences. So what happens when we introduce within these sequences, right? What we cut this target gene sequence using endonucleases, like such a way that it forms sticky ends within the selectable markers region. So when they cloning sites or selectable marker regions where they form these sticky ends, right? So these sticky ends are formed. Once these sticky ends are formed within the, now what happened? So within this selectable markers ampicillin resistance conferring gene sequence, we introduce our target gene. Now, once the sequence is disrupted by our target gene, do you really believe or do you really think that this resistant gene would work? Will it work? Is my question is clear? So in NCERT they given very clearly that how do we clone or how do we really select or how do we distinguish between recombinant and non-recombinants. For example, transformed and non-transformants. So if you see that an E. coli plasmid, normally in general, they will not have these resistance genes, right? But we can prepare a plasmid in such a way that we can use this resistance genes as an markers. So once we introduce our target gene within this selectable marker DNA sequence, right? What happens? The DNA get disrupted. In the sense, the sequence got interrupted by our target gene or alien gene or desired gene. we introduced by the technique of rDNA tech. So this will disrupt. No sir, it will lose its exactly. So it cannot really confer any resistance. Excellent. Yes, Daksha, you're correct. Reminding you didn't get the point. So that's why I'm showing with a model, right? So this is a model. If you think that this red color is a plasmid and this white color regions, right? White color regions as an selectable markers, right? So these selectable markers, within this selectable markers, we are introducing the what do you say? This target gene disrupted the resistance conferring gene sequences. So it cannot really confer any resistance. So based upon resistance conferring gene, okay, now this gene is disrupted by our target gene, right? So now if we provide a ampicillin within the medium and we grow these cells, what happens? Whether they die or whether they live. So based upon that, we can screen if they are dead, right? We can say that they are. We can choose two ampicillins as well. Okay. For example, if they are dead in ampicillin medium, and if they are not dead in no ampicillin medium, it means that they got transformed. In this way, we will use this selectable markers. This is resistance conferring genes. Reminding others, reminding understood, by now you might have very much confident in this topic, right? You guys wrote exams as well. Is that clear? Yeah, good. So this is the main tricky part and very interesting part in this lesson. I really liked it. The transform. How do we really pick the transformants versus non-transformants, right? With this example, we can really understand. Okay. Yeah. So next, coming to the next aspect, that is cloning sites. Cloning sites. Just now we talked about where the ligation of alien DNA is carried out at the restriction site present in one of the two antibiotic resistance providing genes. So once our target DNA, alien DNA, desired DNA is introduced within the sequence of antibiotic resistance conferring genes, right? Those are the cloning sites, right? So that we use, we need these recognition sites, and recognition sites will be provided, or we will be getting with the help of what do you say? These recognition sites will be recognized by the restriction enzymes, and then it will open the plasmid, right? So that we can integrate this piece of DNA, alien piece or desired gene or our gene of interest, right? Once we introduce, we can ligate this alien DNA into antibiotic conferring resistance gene, right? So if you see PBR 322, the diagram is very important, and you need to see that they may give diagram, right? In your academic tests also, you might have wrote about this concept. So in NEET, they may give this diagram, and they may ask you whether what are the sites of restriction, right? Restriction sites, right? For example, you have different restriction endonucleases like HIND3, EcoRI1, BAM-H1, SAL1, PVU2, right? And then you have PSD1, CLA1. All these things, restriction enzymes, I gave it in your worksheet outline of the chapter, right? So these are the restriction sites, and you have to identify that ampicillin resistance gene. For example, this is an ampicillin resistance gene, right? Within the ampicillin resistance gene, within this, you may have the steel is there, na? Let's assume that those are the restriction sites for endonucleases. So this white is ampicillin or resistance gene conferring sequences, and this is the plasmid, within the plasmid you have this selectable markers. Within the selectable markers, what do you have? You have restriction sites for endonucleases. Here it will cut and form the sticky end so that we can ligate our alien DNA. So you need to observe that now you might have seen this plasmid so far in a different way. Now you would have got a little more clear idea, if I'm not wrong. So this is how, now is it clear, this plasmid and how these restriction sites are arranged? Restriction sites are arranged. This, if this white colors are selectable markers, right, like ampicillin resistant genes or whatsoever, and then within that the steel guys are there. Now these are restriction sites for endonucleases. So we cut at this point and then we can ligate our target DNA. Is it clear now more than before? Is it helpful explanation? Are you able to visualize? Are you able to visualize? Yeah. So based, yeah, good. Okay. Okay, let's move. So this is how we can select transformants versus non-transformants, right? So non-transformants usually they have no plasmid. The plasmids which we prepared, right, artificially prepared, right, and then there are other aspects. There are other aspects. Nothing but transformants with non-recombinant plasmid. For example, non-recombinant. They are non-recombinant. It means they are transformants, but they don't have this what is a desired gene. It means they confer resistance to both tetracycline and ampicillin, right? That's what we say, transformants with non-recombinant plasmid. So plasmid is non-recombinant yet, so that's what the other type. The other one is transformants with recombinant plasmid. Once the plasmid, this plasmid is recombinant, this is non-recombinant plasmid, and now this is, sorry, this is recombinant plasmid. Okay, so if a plasmid, if a cell is having which is a transformed as well as which is having recombinant plasmid, it means it will not they are resistant to only ampicillin. It means based upon the selectable markers, right, based upon the selectable markers where we introduced our piece of DNA, maybe we use, there are two antibiotic resistant genes present in PBR 322, right? One is ampicillin and one more is tetracycline, right? So we utilize this tetracycline, right? For example, we use this tetracycline as a selectable marker, and we introduce our gene of interest in the recognition sites of tetracycline. So you may see that plasmid PBR 322, right? So within the tetracycline selectable marker gene is BAM-H1 SAL1, right? So this BAM-H1 and SAL1 will have recognition sites in the PBR 322, and we use those restriction endonucleases to cut our alien DNA specific, our desired DNA or the gene of interest, and we use the same restriction endonucleases to even cut the plasmid at that selectable marker, and we introduce and we form a recombinant plasmid, right? So the other ampicillin resistant gene is not disturbed, so it will confer resistance. So we use tetracycline and ampicillin medium, and then once we use this cells in tetracycline medium, they may die. And if it if we grow them in a ampicillin containing medium, they will live. It means that transformation occurred, right? So this is how we can use this transformation, right? We can select the recombinants versus non-recombinants based upon ampicillin tetracycline mediums. right? So, but this is very tedious process. That's why we choose scientists has gone a bit further, and then they introduced this screening methods that uh, are nothing but the transformants and non-transformants based upon chromogenic substrate. This chromogenic substrate, it means in the regions of chromogenic substrates forming genes, we introduce this gene of interest. So what happens once the gene of interest is introduced within the chromogenic substrate forming, right? um, so the genes which can actually form, the genes would produce enzymes, right? So these enzymes would react with the substrate that we provide in the medium. So enzyme substrate reaction to occur, an enzyme need to be produced. In principles of inheritance and variation also we studied, right? Gene may produce half an enzyme or no enzyme at all sometimes. So here we are introducing our target DNA within those enzyme regions. So enzyme will not be produced. If there is no enzyme, we provide substrate, so it will not form any color. So that implies the white colonies, and if it is producing, it implies the blue colonies. So based upon that, we can select recombinants versus non-recombinants. Is as simple as that. And this mode of selection, we call it as insertional inactivation. So we are inserting our DNA piece, alien piece within the these enzymes, which, for example, beta galactosidase, right? So these enzymes go and interact with the substrate we provide, and it forms the color. Right now, what's happening is we are insertionally inactivating those enzymes, genes, so that they will not produce any enzymes to interact with the substrate to form the blue colonies. So now they will be white colonies in this case scenario, recombinants. If they are not recombinants, they are blue colonies. In that blue white screening is done. That's it. This is very important concept, guys. That's why I took a little time here. So let's is it clear now? Remaining half of the lesson is just step by step process, nothing more concept. This is where everyone would get confused. I bet, I really, I challenge that most of the people would get confused in this topic. Now is it clear? Okay, fine. The next one is we'll be talking about vectors for cloning genes in plants and animals. So these are the this is one of the feature of the plasmids. So we use TI plasmids as agribacterium tumefacients, right? Agrobacterium tumefacients is what we isolate this tDNA, and we use this tumor inducing plasmid to really to transfer our gene of interest into the host cells. So these are the important aspects that you need to cover within the one of the tool that we are discussing is nothing but cloning vectors. And then we will go for the competent host. How do we do the sorry. So how do we prepare this competent cells? So there is a procedure for preparing the competent cells. Once go through the procedure. So how do we, the heat shock method, 42 degrees Celsius heat shock will give for the cells, and immediately will cool down in the ice pack, so that they will open some pores, so that they will take up this plasmid. So this is how we produce the competent host to actually take up this recombinant plasmids. That's how we do. And other aspects we use methods in a more what do you say, in the form of advanced way. We use micro injections. We use a very we can directly inject the recombinant DNA into the cells, or biolistics or gene gun method. Biolistics is a gene gun method, right? We use here microparticles, gold microparticles, tungsten. These are the very important for the NEET questions, right? How do we introduce our target DNA into host cells, competent host, the gene gun methods are very much important. And disarmed pathogen vectors, they infect the cell and transfer the recombinant DNA into the host cell, but they don't damage the host. That's why we say disarmed, right? Unarmed or disarmed, we say, right? So these are the isolation of genetic material. And what are the enzymes that we use in isolation of this genetic material, like proteases, cellulases, lysozymes, ribonucleases, because we need specifically only DNA, not any other macromolecule. So we use different enzymes, right? Proteases to cleave the bacterial cells, and we can isolate them. Chitinases in the case of chitin degradation of chitin. So we add all these things to really purify the DNA. We need only DNA molecule, not any other molecule, right? So first isolation of genetic material, that is DNA. That is the first process. The second process is nothing but cutting of DNA at specific locations. We use restriction what do you say? How do we cut? Right? We use we have to cut the gene of interest, and then we have to identify whether the gene of interest we cut is present or not. So we use agarogel electrophoresis to see the desired genes, whether they cut at or the fragmentation is done by restriction enzymes or not. PCR is very important, and the conditions are important. Like annealing temperatures. What are the temperatures of annealing? Right, extension, primary extensions. Right. At the same time, the functions of Taq polymerase, and where does this thermus aquaticus? If you see this. From where we isolated this Taq polymerases. The Taq polymerases, one of the important aspects, even 11th standard I talked about, right, when we are in first floor, right, what we talked about Taq polymerases, we exploited the very nature of the enzyme that has a stability over high temperatures. So if you see the PCR method, we use Taq polymerases, not our regular DNA polymerases, where they degrade at high temperatures. So we use this Taq polymerases for the primer extensions. So these are the conditions you need to be very much careful about PCR, right? Polymerase chain reaction. Who invented this? PCR, and then denaturing temperature, annealing temperature, and then extension, primer extension, and enzymes required for it. So these are the aspects you need to cover in PCR. And then this is the third step, and fourth step is, once the PCR is done, we insert the recombinant DNA into host cell. We know how to make the recombinant DNA, and then once the recombinant DNA is prepared, the plasmid is prepared, then we know how do we produce? Like we produce at the selectable markers, ampicillin resistant gene sites, and then we transform those cells, competent cells. Right, select those. What we see is we grow them in a large scale. At the same time, we obtain the foreign gene products. So this recombinant protein needs to be produced continuous cultures, and then we can use bioreactors also to produce. Right, bioreactors are concerned. Right, so bioreactors, their functions and their types are very important. For example, what is the difference between a still tank bioreactor with a sparge still tank bioreactors? So these are the aspects you need to focus on. So and then once we get the products, right, then we go for downstream processing. So the downstream processing, such as trials, quality control, testing, etc., would be taken care. So with this, we completed this chapter. So I think this explanation is useful, and you got an insight or better understanding of this concept. I believe in that. So is that okay, guys? We'll move to the next chapter. So these are the hot areas you need to cover. Shall we continue the next chapter? That is strategies to enhance food material, food production. Quickly we'll go through. So we talked about yesterday. We already talked about outbreeding, inbreeding, in different breeding techniques. Hope okay. So when coming to the next lesson, that is the next half of the strategies to enhance the food materials. That is, we use this. We talked about inbreeding, outbreeding, within the outbreeding, we talked about outcrossing, crossbreeding, and interspecific hybridization. So I hope that you need to you already revised the examples still here. So now we will talk about the control breeding experiments. So as I told, right, human work for humans, right, and we always try to use this science for human welfare. So no matter what is the damage we do to other organisms. But let's put in a positive perspective and move ahead in this. That is, breeding techniques, or we will be doing in a controlled setup. For example, what are the control breeding experiments for that matter? Like artificial insemination, in academics itself, I have already explained and shown different pictures regarding how these control breeding experiments would work. So when it comes to the control breeding experiments, first one is artificial insemination, and then MOET, right? And then so if you see these artificial control breeding experiments, right? So which is more, which is more, efficient for that matter, right? From the natural breeding, right? We allow them to breed to very controllably using hormones to produce n number of or more ovums in an desired female animal. So this is a gradation, right? This is start, and this is how from a conventional natural methods to an technique like MOET. So the MOET is more efficient system, efficient experiment, breeding experiments when it comes to the efficiency or increasing efficiency of fertilization, right? So in MOET, you need to focus on why we use MOET, in what cell stage we actually remove, right? These embryos without surgically, non-surgically, we will be removing, and then we will be putting in the surrogate mothers, and then at one shot we can produce 10 different. 10 similar animals, or similar desired animals that we intended for, right? So these are the areas you need to focus in the animal breeding. MOED, its functions, and what cell stage we transfer these embryos, whether surgically or non-surgically, which hormones we use, and how does that affect the ovulation. So super ovulation takes place. So these are the aspects you need to work on. So the next aspect is beekeeping. So beekeeping or apiculture. In beekeeping, right, the Indian most common species you need to understand. You should be remembering the common species we rear, like Apis indica. Beekeeping needs, right? For example, it needs to have a proper management of beehives and handling and collection of honey and selection of suitable location for beekeeping beehives. So if there are no plants and there is plants with no flowers or no flowers at all, so obviously it's a not an area to beekeeping. So these are the aspects you may get in correct or incorrect statements. Okay, in this nothing more important. And then species names are important, so you need to focus on fisheries. Fisheries, the terminology. For example, freshwater fishes, what are those examples? Marine fish, what are those examples? And what is difference between aquaculture and pisciculture, right? So aquaculture, farming of aquatic organisms, whereas pisciculture is farming of fishes. We use fishes term here because we farm different types of fish. Okay, and then you have to focus on the blue revolution. That is nothing but fishery industry. If you talk about white revolution, these are the aspects you may get. What is white revolution? What is blue revolution? What is pink revolution? Pink revolution is nothing but what do you mean by pink revolution, guys? Pink revolution. Pink revolution means what? Wearing pink dresses or pink colored bags and pencils or whatever. What is pink revolution? Yeah, poultry and meat. Correct. Yeah, that's correct. Okay, like that. In this lesson, you need to focus on these revolutions. Okay, that is pink revolution, white revolution. White revolution is means dairy industry, and then you have green revolution when it comes to what do you say, plants, right? MS Swaminathan, right? So green revolution. Those are, and then we talk about blue revolution. That is fishery industry, flourishing the fishery industry. Okay, revolution is not just to start, but it needs to be even flourish in that sense. Now coming to the plant breeding techniques, right? So in plant breeding techniques, we talk about green revolution, right? MS Swaminathan, right? And then we have to talk about even what are the desired traits that we look in the crop, right? For example, increasing crop yield, improvement quality, at the same time tolerance to environmental conditions, or tolerant to insect pests. So these may ask as correct or incorrect statements. Now, what are the steps involved in plant breeding? Even in academics, I already explained steps involved in plant breeding. It starts with the seeds and ends with the seeds. Seeds. It means the seeds we need to collect, the germplasm we need to collect, so that the variability, different varieties and different desired varieties come can collect from different parts of the India or the world, and then we will select those in the sense like first we have to collect the genetic variability. We say it is germplasm, and then store them, and then we talk about evolution and selection of parents as the second step. So once we get the seeds, it's not enough, but we need to identify whether they are growing or not, and then we have to select the parents. So out of 100, maybe 10 may show the desired characteristics, and those 10 parents we need to collect, and we need to again take the seeds of it, and then we have to further use their what do you say, pollen, and a pollen for cross hybridization of the selected parents. Once we cross hybridize, what happens is we produce offsprings, and this cross hybridization has some limitations, right? Like it is very time consuming and tedious process. Hybrids may not combine that desirable characteristics. For example, A, B is there. A has some desired characters, B has some desired characters, right? We crossed it, and we got the offspring. So out of 100%, maybe 50 or 60% or may not get the desirable characteristics. Maybe 10 or 20 or 30% of plants offsprings may show the desirable characters. Those we need to pick. It means we need to select them. So these are the limitations. It is very time consuming to really wait for it, this offsprings as well. But still, this is a procedure we use in a plant breeding technique. And the next one, fourth step, is selecting and testing superior recombinants. Okay, superior recombinants. So once we got the recombinants, and we will test for the yield, whether they are actually really working in a natural setting or not, because even we self pollinate them, and we increase the we maintain homozygosity. We know what is homozygosity, right? The similar genes we try to acquire, or we try to maintain the same genotype over many generations, so it creates homozygosity, right? So once we get this offsprings, we'll go for selection and testing of superior recombinants, right? And then we get this what do you say, now these superior recombinants homozygosity is done, right? Created, it means that they have we got a number of plants with a similar genotype, right? The desired genes we are looking for, so that we will evaluate them. The next and last step is that we will evaluate them in their yield and their agronomic traits of quality, disease resistance. So what we do is, once we get these plants, right, we will test it in a laboratory setting, and then we will shift to actual original farm conditions, and then we will see whether they are working whether they are showing resistance or desired characters, what we see. So likewise, we created a lot of and then once we get once we are sure that they are showing the desirable traits and characteristics, then we go for release and commercialization of these seeds. That's how it starts with seeds and ends with seeds. So seeds of a different variety, and now we have a desired variety, as simple as that. And in India, when it comes to wheat and rice, we have got green revolution in 1960s, and we produced different variety of rice to meet the growing demands of the population food, right? Examples are utmost important, and I gave it in worksheet as well, right? For example, Sonalika, Kalyan Sona, right? IR18, right? What are the different varieties that we produce in which institution? IARI, I told, right, I visited IARI during my degree classes as well. So this Indian Agriculture Research Institute, and if you see this IRRI, that is development at this International Rice Research Institute as well. What they have produced, IR8, if you see these examples. So what are the different varieties that are produced in India and other countries and worldwide? So that's how you need to focus. So who actually, the Nobel laureate who actually developed a semi-dwarf variety? This might be a potential question. Norman E. Borlaug. So this Norman, who is a Nobel laureate, produced this semi-dwarf variety. Like that, we need to even focus on the what are the other varieties that we see in India. Okay. Okay. So these are not only rice. We have produced even millets, hybrid maize, jowar, and bajra. So these are the examples you need to focus in this area. And then coming to the plant breeding, for disease resistance, I already gave enough mnemonics for this tables, right? If you go through those tables by using that mnemonics, even academics itself, you got exact table in your exam. And if most of the people were very happy that we worked out with some mnemonics. So make use of running notes and that I gave in academics, as well as now what I am telling important aspects, you make use of it, and so it will be definitely useful for you. So don't skip these tables. Tables in NCERT are very important when it comes to this lesson. So you need to practice those according to our mnemonics, right? Right. So in this, we'll be talking about even mutational breeding. So what does it means? Mutation breeding is nothing but we use this mutation as a tool for breeding, right? In mutation breeding, what happens is mutation will create using different mutagens like radiation or chemicals. Radiation like gamma rays to produce plants with a desirable characters, and then we will try to explore the varieties. So in this, examples are at most important. At the same time, vegetable crops rich in vitamins and minerals, right? Which plants show this, which plants has got different types of vitamins, iron, calcium rich spinach. All these examples I gave in worksheet, right? That is example for hybrids with improved nutritional quality, right? So biofortification is one of the aspect you need to cover, and the examples within that, right? So hybrids that we use. And then finally, we talk about the single cell protein, right? Single cell protein SCPs, right? What are the SCPs and spirulina, for example? Spirulina, you need to remember this. What is there? Are you guys following, ma? Are you guys following? We almost came to an end. Yeah. Okay. Hope this class is useful. Yeah. So what we're talking about is single cell proteins. So to meet the larger population food demands, right? because of the population. We can supplement food by using microbes like spirulina and uh, yeah, correct. So how can we really differentiate between the protein that we get from in a regular conventional way to the protein we get from spirulina is much different, right? So spirulina protein we can isolate from organisms, and uh, when it comes to other protein in a natural conventional way, it would be very much less. So we can use this single cell proteins. Spirulina should be focusing. Okay, and then coming to the last aspect, that is tissue culture. Tissue culture, the terminology is like totipotency, explants, right? Micropropagation, soma clones, right? So these are the very important terminology you need to focus and you need to understand and remember. Okay, don't remember for the sake of remembrance, you will forget. Okay, so this is NEET exam, so don't just try to fix something in mind, but you try to understand. If any point of time you feel difficulty, unless or until I'm if I'm not most of the time I will be available, but if I have any headaches or something or the other, I may not I will tell, but I may not immediately respond. But in most case scenario, usually I will be responding for any sort of doubts. So in these tissue culture techniques, you have to focus on this totipotency. It means how this one single cell or a part of the plant can really produce a whole plant. I've shown real tissue culture images as well. What is an explant, and where we use this micropropagation? For example, the micropropagation is nothing but producing thousands of plants in a very short time, right, through tissue culture. And what are soma clones? Soma clones of the plants will be genetically identical to the original plant, right? What are the examples, right? At the same time, how can we do this meristematic cultures? For example, if the whole plant is diseased with a virus or something or the other, and still we wanted to save those plants, in the sense like we, by using totipotency, we can produce a new plant of the same kind, right? So how do we exercise, or how do we isolate a disease free tissue? Which tissue we can go with is the meristematic tissue. So because the meristematic tissues, apical meristem, you would have seen during plant growth regulation in 11th standard botany plant physiology. I will explain that as well. So meristem is something which is a rigorously growing and proliferating tissue, so that it will we can exercise that tissue to produce a disease-free plants. And then we have to talk about the somatic hybridization. What is somatic hybridization? We use polyethylene glycol to somatic, to fuse this two different varieties of plants. It means the protoplasms from two different varieties of plants. So to fuse these two different protoplasm, we use PEG, polyethylene glycol. So on that, well, so these are the hot spots and very important aspects that you need to cover. So for tomorrow is Thursday and you have a test, and you can utilize that. Tomorrow there is no subject, particular chapter we will not discuss, right? So if you have any doubts in this following lessons, you send me before 6 p.m. today itself, right? To my WhatsApp number, you can send the topic that you want me to discuss tomorrow. Okay. Otherwise, we can stop the class by half an hour or whatever. We'll see. Okay. Utilize this Thursday for kind of an explanation of topics which you have doubts. I asked several times to send the topics you want me to take up, but still, if you are very much clear with the 12th standard topics, that's okay. I'm very happy about it. But still, if you are hesitating, please don't hesitate to send the topics, so that if you send topics, 1 or 2, 3 topics, we can discuss tomorrow and we can utilize time best. So that the Thursday revision you can do, and 5 to 8 you can write the test as well. So this is the whole intention of Thursday classes. It is a quick revision and doubt session, not the class session. Is that clear, guys? And you have to make use of it. So that is it for today, guys. I hope this session is very interesting and useful for you, especially the biotechnology lesson. And please do like before you leave, and please do subscribe, as well as to share as much as you can share to your friends. If you feel that this is really worth to share, so you please share it so that they also get benefited. Okay, as simple as that. That's why I'm using different models in a workable model. This simple aids, the simple things is readily available at your home also. While you're reading, you can use this to even better understand. Okay, so leave the feedback in the group as well, and I hope you enjoyed this class, and we'll meet tomorrow. Take care, stay home, stay safe. Is that okay, guys? Is there any doubts out there? Please let me know. Okay, you can follow me at my Insta also. You will find a lot of variety of topics in this Insta. Okay, you can follow there as well. Okay. At the same time, I'm creating this links, everything at one place. You can access that this link at any point of time. Okay, yeah. Okay then. Thank you, guys. See you tomorrow. Take care. Thank you. See you then. Yeah. Okay, signing out. Thank you. Take care. If the links are not working, please message me. Okay. Thank you. Yeah. Thank you.